I would like to now give you several more strategies, not to communicate a problem, but to communicate either a solution or the goal of your research in one impactful sentence. Let me share an inspirational quote. Can everyone take a moment and read this? I'm going to tell you something else about me. I'm huge into salsa dancing. I don't know if that came up in your research, Eva, Janine, if that came up in your research. Did it? A little bit? Okay. So in 2010 in Vancouver, I organized the Vancouver International Salsa Festival. 18 countries represented, had about 15,000 participants. And while we were organizing, my business partner and I, Cheyenne, would have all these vendors kept trying to sell us like ticket management, people management, organizational stuff, competition programming. And every single time someone would pitch us, my partner Cheyenne, after about 30 seconds, a minute, would be like, sorry. Can you just give it to me in a sentence first before the details? And if you couldn't do it, he'd be like, thank you very much. You didn't know it well enough. First strategy for you to be able to summarize the solution or the goal of your research or the opportunity you want to take advantage of is with the 24/7 challenge. Have any of you heard of the Ig Nobels? Amruta, you have? The Ig Nobels are one of the greatest things in the world of science. They happen at Harvard University every year, and their slogan is research that makes you laugh and then makes you think. And the way it works is this. A lot of the people, research that is ridiculous, like just but it's real research. It's real, like why don't woodpeckers get headaches? And if you win an Ig Nobel, an actual Nobel laureate presents you the prize. It's a huge thing in the world of like science research. But they have this thing called the 24/7 challenge, where you have to explain a difficult concept or your research in 24 seconds and then in 7 words. A technical explanation, and then an explanation that anyone can understand that will make them curious. I'm going to show you a few Ig Nobels, and then you will do the challenge. I'm going to pair you up in rooms. One person will speak for 24 seconds and then 7 words, and then the next person, 24 seconds and then 7 words to identify the core of what it is you're trying to do. First, some Ig Nobels. This is Rich Roberts, who is a Nobel laureate in biochemistry. He won the Nobel Prize of Chemistry, and he's from biochemist at New England Biolabs.
Rich Roberts.
Yeah, it's ridiculous. It's totally ridiculous.
Clock genes.
Clock
First,
genes.
A complete technical description of the subject in 24 seconds. On your mark, get set, go.
The original circadian locomotive output cycle's caput or clock gene encodes a basic helix loop helix pass transcription factor called clock that is one of a family of genes that control circadian rhythm in mammals. More than 20 genes are involved with such catchy names as period and cryptochrome. The products of many of them are activated on others in success.
Now here's the thing. That is a technical explanation, and that's what all of you sent me in your slides. All of you used a technical jargon, and it's not that it's bad. It's that before we get to that, we need to have something that's going to summarize it or create curiosity. And so that's where the 7 words comes in. Here's the 7 words for clock genes.
Now a clear summary that anyone can understand in 7 words. On your mark, get set, go.
Clock genes are responsible for jet lag.
Now, if you had said that at the beginning, I would be like, what are you talking about? And then you've primed me for your technical explanation. And then at some point to engage Area 47, you explain that. And we all go, oh, and you got me. And if I'm only going to remember one thing you say in a general impression, maybe it's going to be those 7 words. And the general impression was like, that was really interesting. That was it. That was it. You should watch that. Too many great ideas, my friends, are lost to boredom. Adding a few little things like this will make things memorable and impactful. Let's do another one.
And infectious diseases, a pioneer in computational biology. Pardis Sabati. Her topic, viral evolution. First, a complete technical description of the subject in 24 seconds. On your mark, get set, go.
The 2013 to 16 West African Ebola outbreak led to over 28,000 confirmed cases, starting from a single entry into the human population. Over numerous rounds of human-to-human transmission, Ebola virus genome replication generated thousands of mutations, including an alanine to valine non-synonymous change in Ebola's glycoprotein. Based on multiple in vitro studies, this mutation increases infectivity of Ebola in a variety of human and non-human primate cell types.
Now notice, that was actually not that technical. But how many of you listened to the whole thing? I saw you, right? You're like, yep, interesting. Yeah. I'm not saying that what she did was bad. I'm saying observe the way we actually process information. Imagine if she would have said this first.
Now, a clear summary that anyone can understand in 7 words. On your mark, get set.
Viruses can change really fast.
I didn't say go. On your mark, get set, go.
Viruses can change very fast. They're scary.
What an opening. Imagine if that was the opening to her presentation. We're going to do 2 more. This is another Nobel laureate in, I think it was physics. Wait, no, biology.
Medicine, a professor of molecular biology and genetics at Johns Hopkins University, Carol Greider. Her topic, telomeres. First, a complete technical description of the subject in 24 seconds. On your mark, get set, go.
Telomeres are repeated DNA sequences, TTA, GGG, TTA, GGG, etc., that protect chromosome ends. Every time a cell divides, some sequence is lost and telomeres shorten. However, telomerase can come to the rescue and elongate telomeres by adding TTA, GGG, TTA, GGG, etc. So telomeres are in continuous equilibrium, shortening and lengthening, shortening and lengthening. And when they are too short, cells die.
By the way, if you're wondering who that audience is, that's all postgrads and professors at Harvard, because it's by invite only.
And now a clear summary that anyone can understand in 7 words. On your mark, get set, go.
Telomeres, keeping your cells alive since forever.
So adding a little bit of novelty to a technical explanation. Let's do 1 more for funsies, and then you will do the challenge, my friends. So I hope you're mentally inventorying what you're going to say. This is I think so this is the gentleman. He was a Harvard student. He created Soylent. Do you guys remember Soylent? Soylent was a liquid that's supposed to be all you needed to consume in 1 day. It was just for people who don't like to eat and just need nutrients.
First, a complete technical description of the subject in 24 seconds. On your mark, get set, go.
Metabolism can be understood as 2 complementary processes. Catabolism, which breaks down organic matter into constituent matter and energy via cellular respiration, and anabolism, which builds these components back up into useful complexes such as proteins and nucleic acids. Enzymes are the proteins that are keys to these chemical transformations. Fundamentally, metabolism is about controlling the flow of energy which originates in the superhot core of the sun via fusion.
Now, if he had just said that a little bit slower, it would have been something that we hear all the time in presentations. Right? But notice, you're like, yes. It's the illusion of information because it's so general. It's so distance language. There's so many details in it. So before we can get to that, we have to prime our audience.
And now a clear summary that anyone can understand in 7 words. On your mark, get set, go.
Thanks to enzymes, humans are solar powered.
First.
And I want you all to do another moment of self-observation, because I was watching you, not that. And here's what I saw with everyone. During the technical explanation, that's very interesting. And then the 7 words? Okay, I see what you did there. Okay. Listen, 9 out of 10 times, we only get this. Rarely do we get, oh, okay. Oh, I see what you, okay. All right, very funny. Are you ready to take the 24/7 challenge, my friends? No. Okay, good. But don't worry. Listen, don't worry. Here's what's going to happen. I'm actually going to give you 5 minutes. In partners, think about what you want to do. Write it out if you want. Focus on the 7 words. You're going to have like 5 minutes to do this. Also, if you want to go pee-pee, go pee-pee now and think about it while you're going pee-pee, okay? Any questions before we begin? Hey, gang. So when one person goes, the other person time them, okay? Did you, yeah? So have one person go and be the timer. Hey, Helena.
Hey.
So Helena, when one person goes, like when you go, we'll get Eva to like time you. And when Eva's going, you time her.
Okay, perfect.
Okay.
Perfect. Yeah, I'm currently still thinking.
Hello.
Hello.
So listen, just a quick note, like when Linda goes, Natalie, if you time Linda, and when you're going, Linda, you time Natalie. Like, so take your phone and get the timer ready as well.
Yeah.
Nog ett bisschen.
Hey.
Hi.
Gang, just so you know, like, I'm not sure how far, I know you're probably kind of still trying to figure it out and everything, but when, like, for example, Dominic, when Carmen goes, take out the phone and put the timer on.
Okay.
And Carmen, yeah, I know. And stress, calluses, same thing. When Dominic goes, Carmen, make sure you have your timer ready, okay?
So it's just like to really try to this 24/7 challenge like they did it in Harvard right now.
Yeah, homie.
Cool.
Yeah.
Cool.
Hello. My friends. So Janine, when Mara goes, I want you to take out your phone and get your timer. And
Yes,
and
I'm
Mara,
one of the
get your timer so when Janine goes, you can time the 24 seconds. I'm sorry, Janine, go ahead, my friend.
I'm one of the few people that doesn't have her smartphone very close. So
Oh, that's okay. In that case, you just use you can put a timer on Google. Look at you. Just
Okay.
another reason you're amazing. There you go. Focus completely.
Okay, 24 seconds, huh? Oh shit, I put minute.
Immortale, ti sei giusta molto forte.
Sì.
Oh, sorry, now we need to change language.
We
Was that was Italian, I think. Yes.
you jumped in
Dialect.
siblings.
It's a dialect of Italian.
Yes.
It's like Swiss Italian.
Yes, most likely. Yeah. Let's say it comes from very small region where we live. So basically, each own valley has a different kind of pronunciation of certain words. So
And you guys both know that with each other.
Yeah, I mean,
We're siblings.
Oh, you're siblings. Oh, you know what? I just noticed the last names right now because I thought to myself, you know, I think Switzerland is so fascinating because of like the 4 language thing.
Yeah.
Do you know what I'm saying? It's so in the middle of everything. So I think it's a fascinating country. I really want to go there one day. And so I was like, oh, and I'm fascinated that everyone knows like a little Italian, a little French, a little Swiss. I mean, in Canada, it's just like English or French, and it depends where you are.
Yeah.
Because there's some places where people are like, we don't speak English here. And there's other places like, we don't speak French here. And then there's in the middle, like me. All right. So I just wanted to say, before you go on the challenge, make sure that get your phone ready and put a 24 seconds on the timer.
Yeah.
And then I'll leave you alone. Speak anything you want.
Thanks.
Oh, ma sa che approvi un po' mentalmente.
Hello, Kansu. How are you? Can you hear me okay? Yeah, you're muted. Go ahead, turn on your mic for me.
Yes. Yes, hello.
So, Kansu, right now, everybody's in a breakout room doing one of the challenges. So when they come back, you're just going to watch, and then you can jump in with some of the other challenges. I've been recording the session, so if you want for fun, after today, if you want to watch some of the presentation stuff, you know, you can.
Okay. Yeah, that's good. Thank you. Yeah, sorry, I was in the lab and I couldn't leave before, so.
Okay, don't worry, totally. How are you? Welcome.
Yeah, thank you.
I'm actually, you know what? It's a good thing. I'm going to bring everyone back right now. Welcome back, everybody. Welcome back. Welcome back. Everybody, Kansu's here.
Hello.
Because we're all friends. You guys all know each other, right? You all know each other, right? Eva told me that you've all been like, you all know each other. Like in the normal world, you guys all hang out and like, or see each other at school, whatever. You're best friends. So it is time for the 24/7 challenge. This is when you must explain either the outcome, the goal, or the solution of your research in 24 seconds and then in 7 words. First, can I have a volunteer to be a timer? Who's going to be the timer? Actually, oh, Kansu. Actually, that's really good. Am I saying your name right, by the way? Is it Kansu?
It's Jansu.
Jansu. Jansu. Okay, Jansu. Thank you so much. Thank you, Jansu. So Jansu, please get your timer and you're going to be 24 seconds. So keep your microphone on the whole time. And at 24 seconds, just give me one of these. Oh, that woke everybody up, huh? So it'd be like this, and at 24 seconds, boom, that means you're done. Would anyone, I want to hear from someone who has not spoken yet, because everyone's speaking today. Everyone's talking. So who has not gone yet that would like to go? Eva, okay. Eva M. Mers.
Yes, yes. Okay.
All right, 1 second, Eva. I'm going to spotlight you. Actually, you know what? Everybody, cameras off, except for Kansu and Eva, and we'll give her the gibberish applause. So everybody, cameras off. And so all we're going to see, I'm going to spotlight you, Eva. And in the chat, that's it. You already know the deal. Best gibberish applause. All right. Timer, are you ready?
Yes.
First,
okay.
An explanation in 24 seconds. 3, 2, 1, go.
Traditional plankton measurements taken at a weekly or monthly basis are not sufficient to characterize plankton community dynamics truly. We used underwater imaging as provided by a dual magnification plankton camera as a new tool to phenotype and study species dynamics and size distribution in lakes across 3 levels of the food web in situ with high frequency and high spatial resolution.
Not bad, right? 23 seconds. And now, what did all that mean in 7 words?
Hey, plankton, big brother is watching you.
Now, here's the thing, Eva, that's so awesome. It's fun. And listen, you may or may not feel comfortable doing that like in your actual presentations. However, if you would have slowed that first part down, and just said it, you've probably already said exactly those things in presentations, but then you threw in, so it's sort of like having Big Brother, always watching. Then it gives a little bit of delivery and kind of summarizes and makes some entertainment out of it. So that if someone's going like this, if they have their phone, but you can't see it, and they're like, that's really interesting. Oh, okay, I see what you did. And then you get them back. And then you keep going with the rest of the presentation. And then maybe you do it again. So do you remember how I told you about Big Brother? Here's how we're going to do it. And you have like that little theme throughout as a memory schema. You know, by the way, in the chat, does everyone know what a schema is? In terms of neuroscience? Like how the brain structures information? Just give me a yes or a no. Just give me a yes or a no in the chat if you know what a schema is. No? Okay. So here's how the brain actually works in terms of learning. You learn something once and you go through the whole aspect of learning it. For example, a picture of the ocean is not the ocean. The first time you're like, is this the ocean? When you're a little kid and you're like, that's not the ocean. It's a wall with a picture on it. The only way that you don't do that every single time you see a picture of the ocean, or is this, nope, that's not a picture of the ocean. Is because your brain made a schema where they learned that once and then they just assume. So they're like, we're 99% sure that's just a picture. You don't have to look at it. And that's why it goes away. That's a memory schema. When you add this little tagline, it acts as a schema to access the rest of the information if you wanted it to. Well done. Well done. Bravo. Jansu, where'd you go? Thank you very much. Can we have our next speaker, please? Very well. If there is no volunteers, I will volunteer. And I volunteer Amruta. Amruta, my friend. Let me spotlight you. Everybody, can we get a little applause for Amruta in the chat? And Amruta, am I saying your name correctly?
Yes.
Beautiful. All right. Look at all the love. Thank you, everyone, for the applause. Timer, are you ready? 24 seconds begins in 3, 2, 1, go.
Host-associated bacterial community variation are explained by both a genetic and an environmental component. This is also true for Daphnia galeata. We conducted a common garden experiment where we grew Daphnia galeata in a common garden for 5 years and surveyed their bacterial communities. And we found that 8 different clones retained their genotype-specific microbiota at the end of 5 years.
Boom, well done. And now a summary of your research in 7 words.
Daphnia like keeping their bacteria long-term.
Amruta, here's the thing. I mean, again, if you had that follow through throughout the research in your presentation, it just keeps reminding them the moral of the story. Imagine if you did that, if you started with uncertainty. Oh, okay, dopamine spike. Then you summarize the problem with the perfect world. And then you get into the presentation and just when they start zoning out, like, you hit them with something like that. And then you repeat that throughout your presentation. And what do you think they're gonna remember? Oh, that was the long-term bacteria. They like to keep it long. She did the 5-year study, because this is how memory works. First, you'll have that anchor, that schema, and then it'll remind you of the other information. Bravo, bravo, bravo. Let's hear from the other Eva. Serengeti. I'm killing it, aren't I, Eva? I'm not doing a good job.
The second was pretty good.
Serengeti. So
it's very Italian.
Before you begin, I know it sounds very Spanish. Before you begin, here in like whenever someone says anything Italian-y sounding here, we always do this. It's just like, is that like a total stereotype, or do you have
that's
that
a classic?
Stereotype?
And then you only name food because that's the word that you normally know in Italian.
Parmigiani. Sorry. Okay, that's it. I'm sorry. I'm done. I'm so sorry. I'm so sorry. You're going to be amazing. Timer, are you ready?
Yes.
24 seconds begins in 3, 2, 1, go.
Streams and land are linked by material and energetic flows. These flows exist in a balance because what happens in one system will often affect the other. It is thus fundamental to study the quantity and quality of these fluxes to understand whether target ecosystems are healthy or are suffering from cascading effect happening in neighboring systems.
Wow, that was like 22 seconds. Well done. Well done. And now a 7-word summary.
Metric system flows need balance to function.
Good. Now, here's the thing I like about that. Everyone, let's all turn on our cameras for me, please. Everyone turn on your cameras for me, please. Your first explanation was not overly like complex or anything. It was very standard and it was fine. And then your 7-word summary was also very easy to remember, but still bordered the lines of like a nice formal presentation. It's not as like flamboyant as the others, but it doesn't have to be because there's no one right way. But even still, that one summary helped me to understand what you said before it. And it can remind me if you keep repeating it throughout the presentation. Well done. By the way, does everyone see the impact of that? Like, did you observe your own reactions when you were like, Oh, okay, with the 7 words. Did you guys notice that in yourselves? You are an audience. You are an educated audience who understands the topic and even you appreciate it. Because just because I understand the topic and I know all your terminology doesn't mean I can process everything you're saying as quickly as you. Because what you're presenting me took months to get very clear in your head, and you're only giving me seconds during the presentation to get clear in mine. That's the difference.